So now I am thrilled to introduce you to Derek Huey, who is our research partner up at NAU, who is helping us with our arthropod study. So from their flying cousins down to the ground dwelling variety. Derek is a PhD candidate at NAU. He's working with Dr. Hofstetter, and his dissertation is on how seed harvesting ants help structure plant communities. But his overall research focus is on how arthropod communities are structured through biotic and abiotic factors, which factors very neatly into our goals as well. And so with that, Derek, if you want to share your screen.
Cool, thank you for that introduction. All right, 1 second. There we go, hopefully everyone can see that. All right, yeah, thanks for having me today to talk about some very neat work that's come out of the Sonoran Preserve. I'm up here in Flagstaff, but it's a pleasure to be working down there in the Sonoran where it's nice and warm. And yeah, I'll be talking about arthropod communities on the urban edge of the preserve today. So we've been sampling these ground-dwelling arthropod communities. And what are ground-dwelling arthropods? They're arthropods that live on the ground's surface. These can be things like ants and beetles, mites. It's a whole large diversity. And these ground-dwelling arthropods are really defined by mostly living on the ground's surface, and it's whatever falls into a pit trap. And a pit trap is essentially a cup dug to ground level, and a very easy way to sample ground-dwelling arthropods. And this community of ground-dwelling arthropods is really important for all sorts of ecosystem services like trophic regulation of other groups. Many of these are predators or herbivores. Many ground-dwelling arthropods are important for decomposition and nutrient cycling. And they're really important for linking those above ground processes to below ground processes. And you think of things like ants that while they live on the surface most of the time, they also nest and they dig down below the surface. So they really form this link of above ground and below ground processes. And really a huge defining characteristic of ground dwelling arthropod communities is that there's this enormous diversity that is found in them. We're talking things like insects, arachnids, even some land crustaceans like isopods, and then things that used to be insects but now are in their own group, springtails. So there's just this huge diversity, and that you know, we've seen some great talks on bats and butterflies, but you know, that's just that tiny percent of the diversity that's actually there in the Sonoran Preserve. Most of it is going to be in your ground dwelling arthropods. There's just so many species, and so much is unknown about a lot of these species. So it's really interesting that we have this data that's come from the preserve to be able to look at this. And many of you know the McDowell Sonoran Preserve is this unique urban interface environment where water and temperature stress is pretty extreme compared to other areas. So I was really excited to be able to look at some of this data because a lot of arthropod community data actually comes from wetter places and more mild climates. And so this extreme climate that we have down in Phoenix is really interesting to look at some of these questions with. So this previous study that's come out of the CAP project found that urban areas in Phoenix have more arthropod abundance. There's more total numbers, but the diversity is less. And that was just comparing urban areas, so like someone's backyard, to the desert, to pristine areas. But here we have a different question. We're actually going to look at the urban interface. And so our study objectives for this were to compare arthropod communities on the urban edge, right, where that development, you can see there's houses in this picture, right, versus the interior of the preserve. And we also wanted to examine the effects of temperature and precipitation on these ground-dwelling arthropod communities of the Sonoran Desert. So you may be familiar with this idea of an urban edge and how it affects different communities. So A classic example is mammals, right? Deer or mountain lions or whatever are highly affected by this urban edge. It can really change their habitat because even though the plants might look pretty similar, you know, here versus inside the preserve, the effect of the houses being there changes their behavior, right? So these urban edges can really impact the ecology of systems and make them quite different from the interior of the preserve. So we were wondering if this extends to the ground-dwelling arthropod community. Now, I was able to jump on this project to look at the data and analyze it, but this has been happening all the way since 2012, back when I was an undergrad. This was way before I was actually in graduate school. So this is a great data set. There's so few data sets that span this amount of time for arthropods. And basically, this is the only one I know of that spans this amount of time in an arid environment like Phoenix. So this is a great data set that's been collected down here. And what was established are these interior and exterior site pairs. So you can see here, we have in orange sites that are on the urban edge, very close to houses. And then in blue, we have interior sites that are at least 1 kilometer away from the urban edge interface. And so the thought is that these orange sites should be pretty impacted by the urban edge, and the blue sites should be more like control sites that are not impacted, that are on the interior of the preserve. And so this project was established with the Central Arizona Phoenix Long-Term Ecological Restoration, the CAP-LTR project, in coordination with ASU. And again, that goal is to compare those urban edge communities with the interior communities. And we have a great range of sites. I do want to point out that there are 2 sites here, Dixie Mine and Prospector, that we consider both interior sites because there is no urban edge on this side. So these are kind of like a control pair. And I'll come up later in the presentation. But the other 4 pairs are all are proper interior-exterior pairs. And this project was great. It uses a lot of citizen scientists. I got to go down there and go on one of the collection trips pre-COVID. And I got to see all these sites in person. It was really great. Beautiful down there. I love the McDowell Sonoran Preserve. So it's so neat to be able to go experience it and see all these passionate citizen scientists out there helping collect the pit traps. And so at each site, we placed pit traps out there, which are just the cups dug into the ground, and we opened them up for 48 hours. So you open them on like a Friday and collect them on a Sunday, and you take anything that falls into those cups and you put it into a collection cup. That's what's happening right here. And then you have your sample, and then you go back to the lab and you identify anything that fell in there. Of course, that's easier said than done. Like I said, these arthropods are so diverse, so just figuring out which species you have is a huge problem for any pit trap study like this for ground-dwelling arthropods. It's just so difficult to figure out what the insect or arthropod you have is. There's so many options, and a lot of these groups are very much not defined well. It's very hard to key out your specimen and to figure out what you have. Some of them don't even have the work done on them. There's probably new species all over the place that fell into our traps, but it'd be a huge amount of work to even figure that out, right? You'd have to partner with taxonomists and describe the new species. So I bet a lot of the mites that are down there are probably new species to science, which is pretty neat, but also poses a challenge if you're going to try and understand these community dynamics, because it's the identification level is all over the place. So we used this pretty commonly used practice of going down to the lowest practical taxonomic unit. And that's just to say that most of the time we got down to the species level, but when we couldn't get down to the species level, sometimes we're at the genus level, sometimes we're at family level or higher, depending on the group. So I'm going to be talking about LPTUs in this presentation, but you can just kind of equivalent that to species level. That's kind of what this means. Overall, over those 8 years of pit trapping, we collected 25,477 individuals and identified those to 287 unique LPTUs. So that's a lot of diversity and a lot of individuals. And 74 of those LPTUs, and remember LPTUs kind of like species, are unique to a single site, and 40 of those are found at all sites, ubiquitous at all sites. And here we can see on the right, this pie chart just kind of shows you a breakdown of major groups, you know, what the Sonoran ground-dwelling arthropod community really looks like by the numbers. About 30% of individuals are mites, and about 30% are ants, and 20% are springtails. So those are our 3 dominant groups, mites, ants, and springtails. We also have pretty common are some beetles, true bugs in the order Hemiptera, spiders, and bristletails. And we kind of have this others group of things like isopods and what else? There's a few others that we lumped into this others group. This kind of gives you the spread of what this community looks like. And then, so
7-minute timer.
A 7-minute timer.
That's 11 minutes. So that you have 4 minutes left. If you go 4 minutes, there's no time.
Oh, 4 minutes. Okay, I'll try and go quick then. All right, so if we looked at the interior versus exterior site question. There's 44 of those LPTUs, those species that are unique to interior sites, and 48 that are unique to exterior, and 192 that are kind of shared. So there's actually a pretty big shared pool of species with only a few unique to interior or exterior sites. And here's just some of the cool little pictures of some of the arthropods we found down there. Pretty cool. So this was a neat finding. We were looking, when you compare the interior versus exterior pairs. So down here you can see the orange exterior site, the urban edge site, paired with the interior, the green site. And what's happening in this graph is we're looking at the difference in diversity on between the pairs of sites, the edge and the interior pairs. And this graph shows you the percent difference. So for example, Bell and Gateway, about 50% of their species are different when you go from the edge site to the interior site. And you can see that, you know, all of our sites are between that 40 to about 55% difference going from the edge to the interior. The other thing this graph shows is how they're different, right? Because if you take 2 sites, they can either be different because one species gets replaced by another site, or by another species at another site, right? So you go from Bell and you have an ant species A, and you go to Gateway and it's replaced by ant species B. That's called turnover. That's a species turnover. And then the other thing that can make sites look different is nestedness, when a species is lost. So you go at Bell, there's ant species A, and then you go to Gateway, and there's nothing. We just lost a species. And so that means one site is a smaller subset of another site. And so here the dark bands represent the turnover and the light bands represent nestedness. So the takeaway message is that this is mainly driven by turnover. So when you go from the edge of the preserve to the interior, we're seeing this replacement of species. So one species is being replaced by another. We're not losing species, which is good. I'll try and go quick here because I know I don't have much time. So the other big finding was relationships with climate, and this is pretty standard, right? We have all these groups that share positive relationships with precipitation. Phoenix, dry place. So obviously when there's more rain, there's going to be more bugs, And things like spiders, true bugs, beetles, bristletails, these all have positive relationships with precipitation. Here on the right, we have this 2D space of community composition. So this is just a way to visualize the arthropod communities. Each one of those circles or triangles represents one of our sites at one date. And so if they're close together, they're similar in their community composition, similar bugs. If they're far apart, they're different in their composition. Now, two big takeaway messages. One, you can see that exterior and interior sites are pretty mixed. So there's actually not a whole lot of difference between the urban edge and the interior of the preserve in community composition. But you can see the secondary variables mapped on correlate really well. These are correlations with precipitation and temperature. So it's not the edge versus the interior that's driving the differences in arthropod community composition. It's really relationships with climate, things like precipitation and temperature that are driving our arthropod community composition. So hopefully I can wrap up here in a minute. So ground dwelling arthropods, again, similar between exterior and interior sites, the composition is, but there are some important differences of turnover. There are some species replacements. And this kind of contradicts work from other regions and in other communities that show that the urban edge is impactful to arthropod communities. So that's interesting. Maybe here in my second point, climate, especially precipitation, is linked to the arthropod community dynamic. So maybe precipitation is just the overriding factor, and it's such a strong factor that the urban edge isn't as impactful. And with that, I just want to acknowledge a whole bunch of people and many citizen scientists that helped with this project. And I think with that, I'll end the presentation. Thank you for having me.